Hello, everyone. My name is John Asmus, and I did my project on Toxoplasma gondii and trying to knock out the SUB4 gene and find whether it is vital in host cell invasion. So on April 13th, I presented for the National Conference on Undergraduate Research. And so in a few moments, I will play the recording of my presentation for that. And then afterwards, I will show a picture of the poster I made. So let me share my screen. Awesome, so here it is. I'm gonna play this.
So it is my absolute pleasure to introduce John. John, you are welcome to present first. Go ahead.
Awesome. All right, so I'm John Asmus. I go to the University of Washington, do research under Dr. Swati Agrawal. I'm a pre-med student looking to go into pediatrics. And so the research I've been doing is on Toxoplasma gondii, specifically the SUB4 gene. And so I'm looking to delete that and then see if they're still able to invade host cells. So first of all, what is Toxoplasma gondii? Well, it's a unicellular protist, and it's an obligate intracellular parasite, meaning that it has to be inside of a host cell in order to reproduce. It is part of the phylum Apicomplexa, and it causes the disease toxoplasmosis. And while in most humans, toxoplasmosis is relatively harmless, it can have some pretty severe complications if immunocompromised individuals are infected. So that being any patients going through chemotherapy, HIV or AIDS, infected people. Also, infants born to infected mothers can have pretty severe complications such as blindness and severe ocular diseases later in life as well. So Toxoplasma gondii's life cycle, cats are the definitive host, and so it can replicate in any mammal, but it can only sexually reproduce inside of cats. And so then once cats, it spreads from them mostly through their fecal matter, which is why you may have heard pregnant women advised not to change cat litter. This is exactly why, because if they get infected from the cat litter, then their babies can have severe effects later on in life. Also from uncooked meats is the other main way of infection in humans. So as I mentioned, Toxoplasma gondii is an apicomplexan. So these are, it's a phylum of all intracellular parasitic protists, meaning that they have to be inside of another cell in order to reproduce. And so two main things that set these protists apart from others is an apical complex and an apicoplast. So the apical complex, if you see here down on this apical end here, this is where they have a bunch of different organelles such as micronemes, rhoptries, dense granules that secrete proteins that are involved in host cell invasion. So they help attach on to other cells and then invade them. But all these proteins come and they accumulate down here at the apical end of it. And the other thing is an apicoplast, which you can see is right here, this green organelle here. All apicomplexans except for Cryptosporidium have an apicoplast. And what this is it's a plastid that it doesn't photosynthesize, even though it was originated from algae, but it's really important in fatty acid synthesis and other metabolic pathways. So the three most notorious apicomplexans are Plasmodium, Cryptosporidium, and Toxoplasma gondii. And Plasmodium is what's responsible for malaria. So obviously that's like a huge cause of death throughout the world, and it's just very detrimental. And then Cryptosporidium causes cryptosporidosis. And as you can see here in the bottom right corner, cryptosporidosis is the second biggest cause of diarrheal disease and death in infants. So this shows that apicomplexans aren't just parasites, but they're many times lethal parasites. So as I was talking about the apical end here, there are many different secretory organelles in these Toxoplasma gondii, including, as you can see here, these black organelles here are the micronemes, these pink ones here are the rhoptries, and these yellow ones are the dense granules. And these are really important because they secrete proteins that are involved in the adhesion to the membrane of a host cell, and then also proteins that go in and are involved in the invasion of that. So they go through and they actually invade into the cell, and once they invade into these cells, then they can reproduce inside of them. So one group of really important proteins for this are subtilisin-like proteins, and what these are a group of serine proteases that function in preparing other proteins for adhesion and invasion. So basically, these proteases, they process these other proteins and get them into their final form for adhering to these cells and invading them. And so there are 12 subtilisin-like proteases in Toxoplasma gondii, and they're coded for a corresponding set of genes, SUB1 through SUB12. SUB1 and 2 have research done on them. So the SUB1 protein, it is very important for surface processing of microneme's adhesive proteins. So it basically processes them into a final form where these proteins are able to go and attach to Toxoplasma gondii onto the membranes of these host cells that they're invading. And then SUB2 is important in processing the rupture protein 1. And so that's, again, just important for making a final form of a protein that can go and be involved in the invasion of these other cells. But for SUB3 through SUB12, there's little to no research out there on them, other than you can see where they are in the genome and their genomic sequences, but there's barely any research. Honestly, I couldn't find any research on the function of these. And so I wanted to look at the SUB4 gene and see, is this vital for, like, can the Toxoplasma gondii live without this gene? And another reason why this could be really important is because these subtilisins are conserved in other apicoplasts such as Plasmodium. And Plasmodium, which causes malaria, can be really hard to study, but Toxoplasma gondii here will act as a really important model organism. So we can apply this research to Plasmodium and malaria research as well. So my research question was, is the SUB4 gene vital for Toxoplasma gondii host cell invasion? And the methods I'm going to use to answer this question are using a CRISPR-Cas9 gene modification tool to create SUB4 gene deletion mutants in Toxoplasma gondii tachyzoids. And then I'm going to observe their ability to invade human foreskin fibroblast cells. And then I'm also using SAG1, which is surface antigen 1 gene, as a control gene, because knockout experiments have been done many times with SAG1, and so it will be able to see if we're doing everything right. Because SAG1 is, many of these experiments have already been done with it. So the first step is creating primers that will allow the constructs I create to identify the SUB4 gene when put in with the Toxoplasma gondii. So we need to make 6 different ones. 2 of them are going to be up at the 5' beginning area of the SUB4 gene, and then at the 3' end. And these are for the Cas9, which is the tool that's used to make cuts to be able to identify the SUB4 locus. And then 2 other primers we need is 1 upstream and 1 downstream as homology flank primers. And you can see here, this is the entire genomic DNA for SUB4 in the light blue. And then in the black is the immediate upstream and downstream regions. And so here in this pink, you have the upstream homology flank primer that we made. And then in this teal is the downstream homology flank primer. And then we have 2 guide RNA primers. And this allows the Cas9 to identify and make cuts. So 1 at the beginning, 1 at the end of the sequence. And as you can see here, the results, these are the primers that we actually ended up ordering. And here are the sequences for them. So again, we have the 2 homology flank primers. Then we have a forward and then complementary reverse primer for both the 5' and the 3' guide RNA primers. So then the next step was I had to make a construct. And so this was the PJET cassette with homology flank primers. So the PJET cassette is a drug resistance cassette. And I used polymerase chain reaction, or PCR, to amplify this with homology flanks. And so as you can see here, we made a homologous repair template by adding the 5' and 3' homology flanks, basically connecting those to the PJET drug resistance cassette. And then the next construct that we had to make was, or actually, excuse me, I'm going to show the results now. So we successfully made that, and we confirmed it by using gel electrophoresis. And using the APAP, we could see that when combined with the homology flanks, this DNA should have run at 2.6 kilobases on a gel electrophoresis. As you can see here in lanes 3, 4, 5, and 6, it did successfully run at about 2.6 kilobases. So that shows that we successfully made that construct. The next step was creating a PU6 vector. So the PU6 vector, what that is it contains the Cas9 that makes cuts into the genome. And so we had to make a construct. We had to add the guide RNA primers, that way they could direct the Cas9 to make cuts at the specific point in the gene. And so this takes 3 steps. First is annealing. So we had to anneal the forward and reverse primers here to make a double-stranded primer. And then once we had that, we had to ligate it in this PU6 vector, which contains the Cas9. So then you can see here, we would have the sgRNAs with the PU6 together. And then lastly, we transformed it in E. coli cells. And I think this is really like a toxic relationship. We take these E. coli cells and we use it to amplify our plasmid, and then we just kill it and take all of our plasmids away. So it's a really toxic relationship. But I'm still working on getting that to be successful. I have, as you can see here, we successfully annealed the sgRNA primers, as these are the single-stranded here, and as you can see, they run a little lower than the annealed primers. And just this little bit of difference can tell us that these are double-stranded versus these are single-stranded. And here, this is part, this gel electrophoresis is part of the ligation process. So in order to attach those guide RNAs with the PU6, we had to make a cut because the PU6 is circular, so we had to cut it to open it up. That way we can add the sgRNAs on either side. And so this shows that we used the BSA1 enzyme, and it shows we successfully cut it because the linear runs higher than the circular, because circular DNA runs faster than linear. And here are some of the transformation colonies. We later did digests and found out that the PU6 did not successfully uptake the sgRNAs, and so that's something that we're still working on. And I plan on coming back. I'm graduating this spring, but I plan on coming back in the summer because I want to finish this research. I think it's really interesting. So the next step is going to be tissue culture. I've gone ahead and done some of this. So we had to culture both host cells and the Toxoplasma gondii. So the host cells are human foreskin fibroblast cells, which you can see up here. This is a picture under a microscope. And then the next one was we had to grow the Toxoplasma gondii cells. And so what we used, it's a strain called Q80. And what this is it has the non-homologous end joining, the mechanisms. It has the mechanics for it knocked out. That way, our homology-directed repairs can integrate the PJET resistance cassette into the SUB4 locus. And so as you can see here, we actually put these Q80 cells in with the foreskin cells, and they actually invade them, and then they replicate. So this is them with the foreskin cells, and this is when they've exhausted the foreskin cells, and there's just a whole bunch of Toxoplasma gondii right here. And so I've successfully grown those, and then I've frozen them down and have them in -80 Celsius right now for later use. So step 4 of this will be transfection, which we're transfecting the Toxoplasma gondii with the 2 constructs that we made. So first, the PU6 with the guide RNAs come down here, and they make cuts in the beginning and end of the SUB4, and they knock out that gene. And next, we have the homologous repair template that we made. This comes down, and the homology flank recognizes the up and downstream regions. And then the PJET drug cassette actually basically replaces the SUB4 gene. So it kind of like tricks it into taking the drug cassette instead of the SUB4 gene. And next step will be doing drug selection. And so the PJET cassette codes for an ampicillin resistance. And so if the Toxoplasma gondii successfully uptakes the PJET cassette, then they will be able to survive exposure to ampicillin. So this will tell us whether or not whether they can survive exposure to ampicillin. Will tell us whether or not it successfully uptook the PJET cassette. And lastly, we'll do invasion and egress studies. And so this will measure the ability of these transfected cells to invade and replicate in the foreskin cells. And we will compare non-transfected cells, their ability to invade to the transfected cells. So we'll be able to see without the SUB4 gene, is it still able to invade host cells, or is it able to do it as quickly? And so this will give us insight into the viability of, or the vitality of the SUB4 gene in host cell invasion. And lastly, here are my references, and my acknowledgements are, I want to thank Dr. Agrawal. She's been such a great mentor. The whole biology department and donors, and then my girlfriend, Jana, my family, and my friends for their support.
I'd also like to thank the honors program and Dr. Slunt and Dr. Scanlon. They've been a huge part of me doing this research and just being able to do my honors capstone. And so lastly, here's this is a picture of the poster that I made for it. And so most of this was gone over and explained in the video presentation, but if you want to pause it and take a look over it now, you can do that. And so I just want to thank you for watching. If you have any questions, please feel free to reach out to me. And I hope you enjoyed, and I hope you learned something. Thank you so much.